Hello, my dear students. Welcome to this online session regarding open elective paper prescribed for semester 2, first year of Maharani Cluster University, Bengaluru, and the Department of Studies in Zoology under School of Sciences is offering Zoology Open Elective entitled Parasitology. And I am Dr. MEERA B K and my colleague Dr. Lata V. We have prepared some question bank pertaining to chapter 5, 6 and 7 of your syllabus, prescribed syllabus for your paper. And in my last offline class, I have briefed you about the syllabus and the scheme of evaluation and how are we going to complete our sessions as far as this study of parasitology paper is concerned. Now, we will look into these questions one by one and discuss them. Okay? Good. So, the first question in our question bank is something regarding the giving the expansions of few epithets, which are basically the techniques used in molecular diagnosis. Okay? The short form, in other words, is called epithet, E-P-I-T-H-E-T. And the first question says, expand the following epithets. Here I can see 4 techniques, 4 epithets. Number A, ELISA, E-L-I-S-A. B, DNA. C, RNA. D, CCI. Okay. So for your examination, any 4 epithets can be given for expansion and asked for 2 marks, carrying half mark each for the correct answer. Or if the paper needs to be simplified, any 2 epithets can be given, but here we shall consider it as 4 epithets. And in this question, first epithet is ELISA. It stands for enzyme linked immunosorbent assay. Be careful about the spellings, don't write wrong spellings. Enzyme linked immunosorbent assay, E-L-I-S-A. Okay, this is a technique used for diagnosis of the disease. Normally, the blood samples are given to the laboratories. Whenever we go to a doctor, he suspects something and asks us to get our blood samples tested for few parameters, and he will prescribe the medicine after looking at the results. Apart from blood analysis, or for blood analysis itself, some techniques are used. They have been kept under the group molecular diagnostic techniques. ELISA is one such technique. I am going to tell you in bit detail when I take up question number 3. So, question is ELISA. Your answer will be enzyme linked immunosorbent assay. If you want to be very clear, underline the alphabet E, L-I, S and A. So telling the person who corrects the paper how this epithet has been formed, the answer will be more convincing and it will be appreciated. And then B is DNA. Of course, DNA is not a technique. We have cells in our body. Since you students are from different physical science background, I am just briefing the elementary thing. We have cells in our body. These cells contain a central unit called nucleus, and in the nucleus you find this nucleic acid, one type of nucleic acid called DNA, which is going to be our hereditary material. Okay, and DNA is epithet which stands for deoxyribonucleic acid. Deoxyribonucleic acid. So this is the answer expected. Then one more such nucleic acid which we often find in organisms is RNA, that is ribonucleic acid. Okay, it is not deoxy, it is just ribonucleic acid. In most of us, DNA is the genomic material or hereditary material or what you call genes, but in few organisms, RNA also happens to be genomic material. Then the epithet CCI, that is question number D, sub-question number D, stands for counter current immunoelectrophoresis. By now you know that there are certain suppression techniques, okay, like chromatography and electrophoresis, and there are many different types under electrophoresis as well as chromatography. Under electrophoresis we have age, we have page. In this age stands for agarose gel electrophoresis. Like this, counter current immunoelectrophoresis is one type of electrophoresis that comes under separation techniques. I will just let you know what it is when I discuss next few questions. So if you are asked to write about the expansion of epithets, just one line answer in 2 or 3 words, sometimes it can be one word only, you will get your full marks. I advise you to answer these type of questions as there are choices. Hope you have understood this. Then we shall go to the second question, that is again expansion of epithets. So here first epithet CCI is actually the D question, sub question D of question number 1, same counter current immunoelectrophoresis, which I am going to brief you when I talk about it. You have another epithet called CFT, complement fixation test. Okay, for what it is used? Again, it is a molecular diagnostic test. For what it is used, I'll let you know little later. And one more thing, please note, you need not have to know the technical procedures or protocols in detail. Okay, you should just, your syllabus says you should just know what that test is, and more than the technical aspects, you should know for what disease this particular test is used. Right now for this question CFT, your answer should be complement fixation test, C underline, F underline and T underline. Okay. Then epithet C is polymerase chain reaction. So this is again a DNA based technique, okay, often used for diagnosis of infections, diseases, okay, viral diseases, etc. PCR stands for polymerase chain reaction. Just to remind you its applicative importance, during COVID times, the patients were sent to the labs to get the RT-PCR done for their this thing, throat swab, right? This RT-PCR is one type of PCR, reverse transcriptase polymerase chain reaction. Okay, like that there are different types of PCR, and there is one particular PCR which can detect mutation. This is not applicable for infectious diseases, but RT-PCR is to detect the viral infection. That is, we have used it for COVID confirmation tests, right? PCR basically is something like a Xerox machine. It amplifies and makes many copies of the DNA sample, whatever you give for that. Okay. So PCR stands for polymerase chain reaction. So RT-PCR is often used to detect the viral infections in the body. Okay. Then another epithet is RIA, that is radioimmunoassay. Okay. Before I forget, this enzyme linked immunosorbent assay, ELISA, sometimes in some textbooks is given as EIA also. So don't mistake, the question can be ELISA or it can be EIA. Answer is same. Now let's get back to RIA. It is radioimmunoassay, one of the methods of assay. Basically, this was devised to know the quantities of hormones present in the blood, and Rosalind Yale, a lady scientist, was the first person to discover this RIA technique, radioimmunoassay. Okay, so whenever you go to doctor, he asks you to get your thyroid profile check done and so many other parameters, hormonal assays, and nowadays people go for this radioimmunoassay. Apart from checking the hormones, this can be used for detection of infections in the blood. So RIA is an important molecular diagnostic tool, right? So RIA, your answer is radioimmunoassay. Nowadays, they don't use these radioactive isotopes. Okay. Little deviation from RIA has come. They may use chemiluminescence technique. Okay. But that's not there in the syllabus. So if you write the expansions, you will get your full marks. This is how you have to go about answering the epithet type of questions. Now let us see the third question. What is ELISA? Mention its use. For your convenience, the answers are also provided in the question bank. As I told you, if it is asked for 2 marks, how are you going to attempt it? ELISA, first tell, this is an enzyme linked immunosorbent assay. Okay, abbreviated as ELISA or EIA. What it does, it tests, it detects and measures antibodies in your blood. What are antibodies? Antibodies are protein molecules that are produced by your B lymphocytes as an immunological response to the antigens, that is toxin proteins released from bacteria, right? So whenever bacteria, pathogenic bacteria invade your body, there is an inbuilt police department, that is nothing but immune system, and the release of antigens in the blood by the pathogenic bacteria is known, is detected by the immune system, and in response to this, the B lymphocytes, okay, they will synthesize antibodies and to nullify or counteract the effect of antigens. So EIA measures antibodies in your blood. This can be used to say if you have antibodies related to certain infectious diseases. And please note, the AIDS, that is uh, a deadly disease, right? That was first detected or diagnosed, by this ELISA technique. They use ELISA technique for the diagnosis of AIDS virus, that is HIV, that is HIV, okay, right, HIV virus. So to detect viral infections in the blood, normally this ELISA is used. AIDS is acquired immunodeficiency syndrome. HIV is human immunodeficiency virus. Okay. So this is how we are going to answer this. You will tell what the technique is and why it is used. A similar question, but not ELISA, it is RIA. I was telling you. This is an immunoassay that uses radio-labeled molecules. Nowadays, nobody goes for radio-labeled molecules because they are potent carcinogens. Therefore, people have shifted to chemiluminescence. But in RIA, they use radio-labeled molecules, okay, to form immune complexes. It's a very, very sensitive in vitro assay. In vitro means the assays or tests what you do under laboratory conditions, okay. This technique, how it works, it is used to measure concentration of substances, usually measuring the hormonal concentrations, and it makes use of antibodies. It makes use of antibodies and it will find out the antigen concentrations. This is how the technique works. And if this question is asked, what is RIA, and mention its use, this would be your answer and this should be fine for 2 marks. Okay. Then let us take up this complement fixation test, CFT. It is a classical laboratory diagnostic test, It is used for determination of virus antibodies in the serum of patient. Serum is blood minus the cells, okay, blood cells. So to detect antibodies in sera of patients, serum singular, sera plural, or to detect virus antibodies, viral antibodies in cerebrospinal fluid, that is fluid that's found in the nervous system. When there is an acute infection, this technique is used, and this normally measures IgG antibodies. We have different types of antibodies that are produced in our body: IgA, IgM, IgE and IgG, 5 types. And each one has different function. Like IgE are concerned with the allergic reactions, and this test measures about IgG antibodies, which are basically concerned with infections. Okay. And next question, let us see. This is not a technique, but often we use these small segments of DNA or RNA segments, which are complementary sequences in DNA or RNA molecules that allow the identification and isolation of specific sequences from an organism. So these are molecular probes, and some molecular probes are readily available, okay, in laboratories. They can be used, and basically they work by hybridization tests, hybridization of nucleic acids, okay. So the probes which involve DNA are called DNA probes and RNA are called RNA probes. And these probes are largely made use of DNA fingerprinting technique, okay. But they find use in diagnostic techniques also, okay. We will go to the next question. CCI. I was telling you, CCI stands for countercurrent immunoelectrophoresis, and it is a very rapid sensitive method for testing or detecting pneumococcal capsular antigens in sputum. So pneumococcal infections are normally are contracted in lungs, and so if the sputum is subjected to this test, you can find out these capsular antigens, and this is a very rapid test. Result can be obtained within 45 minutes. So this is the significance of CCI. Okay, it's a type of electrophoresis, countercurrent electrophoresis. And PCR, I was telling you, this is DNA Xerox machine, to put it in a layman's language. So it allows amplification of DNA fragments even with tiny amounts of biological samples. Okay, like 1 or 2 drops of your tears, or a small drop of semen which has fallen on the petticoats and undergarments of rape victims, or just hair follicles in case of murders, like that, and oligonucleotide primers derived from sequence data. Okay, they make starting with tiny amounts of biological samples and oligonucleotide primers derived from sequence data. This technique is quite fast and easy. So this has taken the DNA technology, which was confined to the research laboratories, to the routine laboratories. Okay, it is PCR technique or PCR machine. PCR technique is done in a machine called PCR machine. And nowadays PCR machines have become like your laptops. What is a laptop to a computer person? Like that, PCR is some instrument for a biologist or for laboratories. Okay. Then name any 2 advantages of molecular diagnosis that we will see. Yeah. What are the advantages of molecular diagnosis? And there are disadvantages also. And here we have given a little elaborate answer. This is too much for 2 marks. We have just explained, okay, sensitivity, specificity, then turnaround time and applications. So if you just say, sensitivity is one advantage, specificity is another one, turnaround time is the third advantage, application is fourth advantage. If you just mention the subheadings, it should be enough for 2 marks, but still we have given a little elaborate answer. Sensitivity in the sense, the tests are quite accurate, and even if the samples are present in very low concentrations, you can get very accurate results. Specificity also, at specific molecule of interest, you can perform this test, and the time is quite less compared to standard traditional culture methods. Okay, and applications, they can be found with molecular methodologies such as infectious diseases, genetic testing, forensics, drug resistance, and tumor marker detection and monitoring. So, the 4 advantages of these molecular diagnostic tests are: number 1, sensitivity; number 2, specificity; number 3, turnaround time; number 4, applications. Okay. At the same time, it doesn't mean that there are only advantages and no disadvantages. There are disadvantages. That is our 10th question. Name any 2 disadvantages of molecular diagnosis. One is very high cost. They are very costly, and all Tom, Dick and Harry cannot do this test. A fully learnt person, it requires lot of expertise. Otherwise, results may go awfully wrong. And you need very specialized instruments, well-equipped laboratories to have accuracy of this molecular diagnosis. Hope, my dear students, here to now we have discussed around 10 questions, and you have enjoyed these sessions. You get back to me over your WhatsApp group. If you are happy with this, we will study some more questions, discuss about some more probable questions of this question bank in our offline class. Thank you so much for your patient listening.